Okay, so again, thank you again for an invitation to talk. It's been really a very nice workshop, and I think testifies to the fact that there is a need for more of those workshops that are informal. And there is a proposal for a KITP workshop in Santa Barbara that hopefully will be successful because we need more time to keep on discussing. Okay, so a lot of the focus yesterday has been on the substrate, right? So there are sort of two sides of an arrhythmia. There is the increased dispersion of refractoriness and discordant alternance being a mechanism to enhance dispersion of refractoriness. But that by itself, unless you have a premature stimulus or a sequence of stimuli, is not enough to trigger an arrhythmia. And so triggered activity is mechanism for occurrence of PVC, spontaneous action potential. It's present and manifested in the LQT syndrome at the organ level, sort of torsade de pointe arrhythmias, which is typical. And so this is, I'll be presenting results of a very, very exciting collaboration that was funded by the NIMBioS Systems Biology Multi-PI R01. So this is a collaboration with Gideon Coren's group at Brown Medical, and they have a very nice animal model, which are transgenic rabbits. So people have studied transgenic mice for years, but they have a short triangular shape action potential. The rabbit has a plateau phase of the action potential, and really those rabbits die with very, very similar pathology as humans. So for example, female rabbits have increased sudden cardiac death postpartum due to hormonal change. This is recapitulating rabbit just as an example. So most of the modeling has been done by Colin Rees, who is a very bright graduate student in my group. Was a postdoc, Jonathan Stoll, helping a bit for a year. And then in Gideon Coren's group, most of the experiments, I'll be talking about cellular mechanism of triggered activity was done by Dimitri Terentiev, who is a very talented experimentalist. So sort of broad picture of triggered activity sort of takes two forms. You have a normal action potential, and you have early after depolarization, which is can occur early in the plateau, or typically a little later in the plateau. You have these oscillations that can be sustained or sometimes terminate. And then you have delayed after depolarizations, which is after the action potential is terminated. You have a bump here, which can sometimes just be just a small voltage deflection, and sometimes trigger another action potential. So the dogma is that EADs are due to reduce repolarization, okay, so to prolong the action potential duration, and you allow sufficient time for the L-type calcium current to reopen, because there's a window current I'll discuss on the next line, while DADs have been classically attributed to abnormal calcium handling. Spontaneous calcium release drives the NCX calcium exchanger in forward mode, depolarizes the membrane, and when the membrane gets above the threshold for activation of sodium, you can get a spontaneous action potential. So these have been the sort of picture. There's a very nice work by the UCLA group that validated the importance of the L-type calcium current in the generation of EADs. So these were normal rabbit myocytes. It's published in 2011. Normal rabbit myocyte in which EADs were induced by adding oxidative stress, H2O2, and also by hypokalemia, reduced extracellular potassium. And what they did is they tested the propensity to EADs by changing the inactivation curve of the L-type calcium. So this is the steady state activation and inactivation curve of the L-type calcium current, following the standard Hodgkin-Huxley formalism. So it's a voltage dependent. They're also calcium dependent inactivation. And so there's a window current, which is where these two curves overlap. You can get a current which can be typically around minus 20 millivolts. And so here, what was shown is that if you increase the window current, so here you have If you have the black curve instead of the red curve, you have more overlap. And so you're getting reactivation of the L-type calcium current, giving you EADs. However, if you shift this curve to the left, you reduce the window current, and you can abolish EADs. And this was done in very nice experiments where they use a dynamic clamp technique in which the voltage of a real myocyte is being injected into a computer to calculate a virtual L-type calcium current in which you can shift the inactivation and then re-inject the current into the myocyte. So you have a real myocyte talking to a computer-generated current, but that's a way that you can actually change the inactivation kinetics with this virtual current and show experimentally that the same effect gives you EADs. So clearly, the L-type calcium current is an important actor. So the study I'll discuss was looking at mechanisms of EADs in the long QT syndrome. So there are, I think, about 13 mutations to date. So this is a channelopathy. It has an incidence of about 1 in 1,000, 1 in 2,000 people in the population. That's still a lot of people when you look at the overall population. And the most prevalent are the long QT type 2, which is a loss of IKR, and long QT type 1, which is the loss of IKS. They represent about 30%, each one, about 30% of all long QT incidence. And so there are transgenic rabbits in Gideon's lab of long QT2 type and long QT1 type, and I'll be discussing today only long QT2, which has loss of IKR, so you have reduced reproduction. So this is here in isolated myocyte experiment, leader-made control, normal rabbits, and the long QT2, you have prolongation of the action potential, which in the electrocardiogram would give you a QT prolongation. Mm-hmm. Yeah, so, so it could be that ITO is changed a little bit. It's not going to be a very important actor in what I'll be discussing. And, it's very interesting because we get, what's very valuable, we get many cellular recording, right? So they do 12 cells or 20 cells. And that would be actually better to show that in a talk, the variability that you have in the recording when you really look at 12 cells. And I think that's something that should become more common when experimentally is presented. But you will see that there is variation of ITO. Actually, I'll have a slide maybe at the end that I can show of that in a different talks. But basically, I don't think this is necessarily very significant. But the notch is different. But important and reproducible in many myocytes is that the calcium transient is different here. You can see that the leader-made control, sort of the calcium transient is shorter, and here it tends to be prolonged. Okay, so there is a remodeling of cardiac of calcium homeostasis. So in terms of modeling, if you just take a mathematical model and you take out IKR, you don't get EADs. Okay, so I think this is something very important from a systems biology point of view. Is when you have a mutation, as opposed to doing pharmacological long QT. Pharmacological long QT, you can put an IKR blocker and you will see certain things. But here you have actual rabbit that have developed with the mutation. And because of the loss of IKR and the prolonged action potential duration, as a compensatory change, there is a remodeling of calcium homeostasis. And so this is a first major finding. And so this was characterized. You can take this long QT myocyte and you can do what's called a permeabilized myocyte experiment, in which you put surfactant, you put holes in the membrane, so you're destroying the sort of sarcolemmal membrane, but you flood, you still have all the EC coupling machinery, the calcium handling machinery, the calcium uptake and release, and you flood this into a solution of calcium with calcium buffers, so you can maintain the calcium concentration. And you look in a confocal image, so this is here along the myocyte, 40 microns along the long axis of the myocyte, and this is time. So this is a space-time plot, and you see the calcium sparks that Johannes mentioned in his talk yesterday. And you can count the spark for the leader-made control in long QT2, and what you find is that the spark frequency is increased in long QT2, and so is the spark amplitude. So the ryanide receptor calcium release channel function is remodeled. And further studies that I won't describe in details show that this is due to hyperphosphorylation of the channel. through the CAMK2 pathway, the calmodelin calcium-dependent protein kinase 2, type 2. And so you have hyperphosphorylation of the channel. And this hyperphosphorylation also affects SERCA function, which is increased. So you can also look at the luminal calcium. And so when you increase calcium, starting from 0 calcium, you see that the rate of uptake in long QT2 myocyte, the rate of uptake in the cytoplasmic reticulum is larger. So the time constant is actually shorter. And you can do another experiment to look at the leak. So this is also another measure of the hyperactivity of ranitidine receptor. Is you can put tapsigargine, which blocks uptake, and you can see that actually the concentration in long QT2 myocyte decreases at a faster rate than leadermate control, showing that SERCA is also remodeled. Interestingly, the remodeling, when you do Western blot of ranitidine and of SERCA and phospholamban, which is the inhibitor of SERCA, there is no change of protein expression level. So it's actually accessory proteins that play a role in the phosphorylation that are remodeled and that are changed.
But
yeah.
What about the protein that calcium channel itself? Did they see any change?
They saw the change in the phosphorylation state of the channel.
But not the number of channels?
No, the number of channels in Western blot is the same, but it's the phosphorylated state of the channel. It's hyperphosphorylated, yeah. Okay, so now here are the results of what happens to, so long QT2 myocyte, you saw in the earlier slide, you had prolongation of action potential duration, but no EADs. And when you put isoproterenol, which mimics the beta-adrenergic pathway, then you have, you generate EADs. So these are now the long QT2 myocyte with, so this is a typical recording, in, fraction of the cell, you'll get EADs. And you have the calcium transient is actually, you see you have this sort of prolonged calcium transient. And interestingly, the calcium transient amplitude with ISO is decreased. And so you have actually low calcium, and so it's a little bit puzzling because if you think that calcium remodeling is involved in arrhythmia, why would a lower calcium drives EADs? Because the dogma is that to drive the NCX current more in forward mode, you need actually a larger calcium transient. So here you have a reduced calcium transient. But to demonstrate that the ranitidine receptor remodeling is actually a mechanism of EADs, you add to the ISO KN93, which is inhibits CAMK2 activity, and therefore reduces the hyperphosphorylation of the channel and bring back the ranitidine receptor activity to sort of normal state. And this abolishes the EADs, and of course you also get a larger calcium transient. So this was really the puzzle for modeling. You have EADs with a lower calcium transient than with a stabilized ranitidine receptor. Okay, so please feel free to ask questions at any moment if there's something you need clarification. All right, so we try to understand this phenomena, and using sort of Mahajan or sort of a wholesale OD model of the rabbit myocyte, we just couldn't reproduce this result. You try various parameters, you make RL leaky, but it just we couldn't reproduce this this fact. So we had to go to a more detailed model. So this is just to show you again the confocal line scan. So this is a long QT2 myocyte with ISO. And you see that you have a lot of late calcium releases here during EADs, while when you stabilize the ranitidine receptor calcium release channel activity, by inhibition of CAMK2, you just get a normal calcium transient. So clearly you need a type of modeling that can describe this calcium handling. So we used a model that was developed by Juan Gabriel Restrepo, who was a postdoc in my group. The model was developed in 2008, and it has evolved with further refinement. It's a next generation of the model that was described yesterday. it basically takes into account the fact that calcium is released from diadic junctions, which are spatially distributed throughout the cells, and as a full Markov description of the L-type calcium channel and ryanidine receptor. So it's a similar model that Johannes used in his study of alternance. Use that model right here. Yeah. So the further refinement of making the buffers more realistic and so on, but the same principle. So this is here the T-tubular network, which is imaged. So it's actually the T-tubules penetrate inside the cell, and you get calcium sparks from a unitary calcium release event. So this is so network 20,000 to 100,000 diffuse calcium release units, with a few LCCs and 50 to 200 IOR channel per CRU, Markov model of LCC and IOR gating, and bidirectional coupling between calcium and voltage through a set of current of sarcolemmal membrane current. Okay, so this is just a little simulation of how the resting cell looks like. You have calcium spark. There's one actually, it's a long-lasting spark. Then you have a pace cell. So what is shown, this is a paraview, so it gives you 3D imaging of the cardiac myocyte, which is a sort of a slab here with dimension which are longer along the long axis of the myocyte. And in red, bluish is low calcium concentration, reddish is high calcium concentration. And so this is a normal action potential. So you get calcium release and reuptake in the SR. So this is how the modeling results look like. You have the voltage here. You can look at the voltage. You can look at the calcium transient, and you can do a confocal line scan equivalent and look at the calcium activity, subcellular calcium activity. Okay, so we modeled the hyperactivity of the ryanidine receptor channel. I'm not going to go into the detail, but basically the ryanidine receptor in this model is a four-state model in which calsequestrin can be either bound or unbound to ryanidine receptor tridine junctin, which forms a protein complex. There's a lot of work from Georgi's group, and Dimitri Terentiev, who did those experiments in Georgi's group before, showing that calsequestrin acts as a brake mechanism and gives refractoriness, is responsible for the refractoriness of the ryanidine receptor. So by changing the rate of channel from the closed unbound, calsequestrin unbound state to open unbound, and from closed bound to open bound, you can actually change at the level of a whole calcium release unit the time that it takes for the calcium release unit to recover after a spontaneous release event. And also you can change the spark frequency, and you adjust the model parameters at these channel levels to match the permeabilized myocyte recordings of spark frequency and spark amplitude. Okay. So, you know, at this model, the model becomes quite complex because you can do a simulation, and actually, when you do a simulation, you reproduce the experimental phenomena. But that doesn't tell you necessarily that you understand what's going on, because the model has a complexity which becomes, but at least you can do interventions and look at various aspects of the model. So when we simulated this model, it was very nice because we reproduced EADs with not too much trouble. We really had a hard time getting in a whole cell model. So this is showing you with hyperactive RYRs, which have shortened refractoriness and increased spark frequency. You see the sparking, and then you see that you have late calcium releases. The movie is the entire trace here. And then now you have this sort of oscillation of calcium activity, because of course once you get the EADs, ICA reopens, and so you recruit more sparks through normal EC coupling, calcium-induced calcium release. But the key mechanism is this. So if we play the movie again, right? So we have first calcium-induced calcium release, and then you get the late releases, and then you get the EADs. Okay. When we use these models and we used ryanidine receptor with normal function, so normal spark rates and spark amplitude, then we did not get EADs. So this is the equivalent in the model of inhibiting CAMK2 activity with K93, so you stabilize radiating receptor. See, you still get a number of sparks, but you have, that's right, you repolarize. Okay, so this is to show you that we get something similar to the experiments, long QT2 myoside plus ISO plus hyperactive RYRs. You get these late aberrant releases, and then when you stabilize the RYRs to come back to a normal state, you have a more normal calcium transient. Okay, so now the advantage of the detailed model is that we can go and dissect the currents and try to understand what is the mechanism. Because again, the puzzling fact is, from a wholesale perspective, is the following. So this is, this is with normal RYR activity, long QT2 myoside, which is space, this is in the model, and this is with the hyperactivity of the channel, you get EADs. The pacing actually is very slow, both in the experiment and the modeling. To elicit EADs, you had slow pacing of 0.25 hertz. So here is the wholesale calcium transient in the case of stabilized RYR. So you get actually a larger calcium transient, and with hyperactivity, you get a smaller calcium transient. Why is the calcium transient larger? It's because you have, if you have stabilized normal RYR, you have less calcium leak, right? So you have less calcium leak, and so your SR load is larger. This is the SR load, which were not measured in the experiment, although it would be possible to also measure the load at the same time, but it was not measured. But from the lower calcium transient in the experiment, we believe that it's the same as in the computer modeling. The SR gets depleted by the increased spark frequency, late aberrant releases, and so you have a smaller calcium transient. Plotted here is the two traces, so with hyperactive and normal RYR. And so then you can look at the key currents that are participating and are giving you the difference between going into EADs or not going into EADs. You can look at the L-type calcium current, very similar. It gets inactivated by release of calcium. And in the case of no EADs, it terminates. In the case of EADs, it gets reactivated, so you see the L-type calcium current. But the point is that the L-type calcium current difference between the two cases is not responsible for the divergence of those two traces. You get a little bit of difference of IKS. IKS is activated and is calcium dependent. But actually, you get a little more IKS in the hyperactive case, which is actually a repolarizing current. So the real key difference here is NCX. So first you have the, a fast period where you have the reverse and forward mode. But here it's all in forward mode. So this is during this portion of the action potential. In the hyperactive case, where you get EADs, you have a significantly larger NCX current than in the normal RYR case. And so this is very puzzling, because if you have a larger NCX current, how can you have a larger NCX current with a smaller calcium transient? And if we look in the 100 to 300 millisecond, in fact, the cytosolic calcium, the two traces are actually very close to each other, so the wholesale calcium concentrations are very similar. So I'll take any question now. Do you understand what the puzzle is within the standard EC coupling? Yeah, so you have very similar calcium trend, but you have a larger NCX current. Okay. So this is so what we did is we changed the level of hyperactivity a little more continuously between normal RYRs, and the hyperactive case I showed you before corresponds to this blue curve, and these two sort of violet and green curves are hyperactivity in between. And so you can look at the submembrane calcium, because the NCX current is being driven by the calcium in the submembrane space. And here you see something similar, is that in the stabilized case, you have higher submembrane calcium, but again during this sort of critical time period in which the voltage re-enters minus 20 millivolts, so the window for the L-type calcium current to reactivate, the submembrane calcium is very similar. If you look at the release, so this is the total wholesale release current from the calcium release channels, you see that in the case where you get EADs, there's a little bit more release current. But then when you look at this green-purple curve, you see that actually they have the same amount of release current, and one goes into EADs and the other one repolarizes. So it seems like the wholesale release current is a little larger in the hyperactive case, but the submembrane calcium driving the NCX is similar. So the answer to that puzzle is on the last slide. Almost the last slide. Okay. So, the thing is that what's driving the NCX in the myocyte, once you get a level of a detailed model, is not the wholesale submembrane calcium. It's the local calcium which is felt in each calcium release unit. Right? So what you have to look is at the spark rate in conjunction with the concentration dependence of the NCX current. So this is the NCX currents plotted in the range of voltage of interest, plus 20 to minus 20 millivolts. Minus 20 millivolts is sort of important, 0 to minus 20, because this is when you re-enter the window current for ICL to it. So this is the NCX current, and the dashed line is just the part due to the fact that you exchange 3 sodium plus for calcium 2 plus. So this is just due to the Nernst potential, okay? So this is just the electrogenic part. But you also have a mobility part. So this is the calcium from the Bayer's textbook, okay? And so the actual current are those curves here, because as you increase the concentration in the submembrane space, right, there's so many calcium ions that the channel can carry through. And so you have saturation of the current due to sort of the constraint of the mobility of how many ions you can pass. Okay. And so now this is here the calcium submembrane for normal RYR all the way up to hyperactive. And so what you see is that in the normal case, you have larger submembrane calcium. So you see here it goes up to 7 micromolar. In the hyperactive case, you only go to 3 micromolar, okay? So you have larger calcium. But now, how come the total calcium, the wholesale calcium, is the same when you have actually larger submembrane calcium? And that's, you have to look at the spark rate. So we measure the spark rate during an interval of a few hundred milliseconds. And it's bin. So this is, you get the spark rate within a bin of a few milliseconds. And the important part is during this time period when the voltage re-enters the window current. So I've blown it up here. And so you have the case that gets EADs as a much larger spark rate, which is normal because you have increased spark frequency, you have remodeled ryanodine receptor. And the case of normal RYR, you have a lower spark rate. So the point is that you get the same wholesale submembrane calcium, or very similar wholesale submembrane calcium, because you have a lower spark rate, but larger sparks, and which are driven by larger submembrane calcium. But in terms of driving the NCX now, how much NCX current does the larger calcium drive? So you have now to go along these curves. And you see that if you have 8 micromolar, right, 8 micromolar at minus 20 millivolts, you're going to get a current of around minus 1.5, while for 3, right, you're going to get around minus 1.2. So because of this saturation of the NCX current with calcium, this difference of calcium does not translate into a very large difference of NCX current. So basically, imagine as an extreme case, to simplify the story, imagine that this curve would be flat. Okay? So if this curve would be flat, it would mean that larger submembrane calcium gives you the same NCX current as a lower submembrane calcium. And so if you have a larger spark rate, right, then you get increased NCX current. And that is the explanation of what's going on. And so we're going to do further experiment to actually block NCX and try to see that. To show that this is a more general mechanism, this sort of a crosstalk of calcium and voltage and genesis of EADs, experiments were also done in leader-mate control, in which ryanidine receptor activity was potentiated by caffeine. So this is these two parts of the slide I didn't discuss earlier. So these are leader-mate control plus ISO. So you get a larger calcium transient with ISO. So this is us when we go running. Okay, sort of larger calcium transient. And then when you add caffeine, you render ryanidine receptor more leaky. You decrease the calcium transient amplitude, but you can potentiate EADs by the same mechanism of increasing the spark frequency and therefore increasing the heterogenic current. And the last slide I'll show you is the validation of this with the modeling. So we put back IKR. We have leader-mate control myocyte. We add the analog of caffeine by making RYS hyperactive. Again, CI is very similar, but you drive a larger NCX current through the late releases. Okay, so there are sort of three messages here. The first message is, of this study is that when you look at an arrhythmia in the context of disease, you cannot just look at one defective channel. Okay, so here in this case, IKR is defective, but you have a whole system-wide remodeling. It's normal because you know the rabbit is living with an increased action potential duration, and it wants to maintain calcium homeostasis, so it's going to have an adaptive response. And so you need to look at the system as a whole system. In terms of second point is in terms of mechanism. This is a case where the detailed models become important because here what was key is to understand how can you get larger NCX currents with a lower calcium transient, which is really contrary to the dogma of calcium overload, larger calcium transient driving NCX. And so this is really, you have to get it down into the details of dissecting what the current is doing. And when the summation of discrete release event, the summation of NCX current, you cannot get in by looking at the whole cell calcium because of the saturation effect. It's actually a nonlinear collective effect. And then the third, I forgot, so I'll just stop here. Okay, all right. All right. Take any question.